Have you ever wondered how microbiologists grow invisible organisms and identify infection causes? Today we explore one of microbiology's fundamentals, bacterial culture media. This topic is essential for exams and forms the backbone of diagnostic microbiology and research. We will define culture media, show main types, demonstrate inoculation and colony growth, and explain clinical relevance. Let's begin with a clear definition. What exactly are bacterial culture media? In simple terms, culture media are artificial environments that provide nutrients and conditions for bacteria to grow. Media supply carbon, nitrogen, minerals, vitamins, and water and control factors like pH and oxygen availability. Media come in different physical forms: solid plates, liquid broths, and semi-solid media for motility testing. Common components include peptones, salts, glucose, blood, or serum when enriched growth is needed. Solid media use agar, a stable gelatin-like agent that solidifies the plate while remaining biologically inert. Understanding these basics helps you choose the right medium for isolation and identification tasks. Media are classified by function. First, selective media, which suppress unwanted microbes while allowing target organisms to grow. Second, differential media, which show visible differences between organisms, often by color changes or hemolysis patterns. Third, enriched media contain extra nutrients like blood or serum to grow fastidious bacteria that need special factors. Common examples include MacConkey agar for gram-negative selection and lactose fermentation differentiation. Blood agar is both enriched and differential, useful for detecting hemolysis patterns that help identification. Remember, selective narrows growth, differential reveals features, enriched supports fastidious organisms. First, prepare media by mixing components accurately according to the recipe and dissolving agar when needed. Next, sterilize by autoclaving to remove contaminants, then cool to a safe pouring temperature for plates. Pour agar into sterile Petri dishes, allow solidification, then label each plate with media type and date. For inoculation, use a sterile loop or swab to transfer specimen onto the plate using streaking techniques for isolation. Incubate plates at the appropriate temperature and atmosphere, then monitor for colony appearance over time. Watch step-by-step colony growth. Small microcolonies become distinct visible colonies suitable for further testing. In clinical labs, media enable isolation and identification, guiding diagnosis and appropriate antimicrobial therapy. Media also support antibiograms. Testing isolates against antibiotics to determine susceptibility and resistance patterns. In research, tailored media allow growth of specific strains for genetic, metabolic, and vaccine studies. Safety is essential. Use aseptic technique, proper disposal, and containment to prevent contamination and exposure. Quick review. Culture media provide nutrients, come in types like selective, differential, enriched, and enable diagnosis and research. Thanks for watching. Keep these points handy for revision and return to the lab with confidence in using culture media.